Well, thanks for being here. It's nice to be sort of presenting a bit of the work that we do on drought adaptation and climate change adaptation. I guess I'm happy enough to work with a bunch of very enthusiastic people in my group, so I would like to thank them at the onset of that presentation. Today we'll be talking about five parts. I mean, first, a few things about drought and climate change, basically sort of basic principles and how to work with it. Second part is going to be on what we have learned so far after some years of working on that constraint. We'll be talking later on a bit of an upstream sort of approach where we go back to the basic biology of drought adaptation, so trait dissection and mechanisms. Then how we use the knowledge that we have gained into breeding application, how do we measure, how do we phenotype those traits that we are working on. And the last part is really about how we link all those pieces together, because we work at different sort of level of sort of biological integration, but at some point we need to make the connection between everything. It's nice to have plenty of genomic resources, but it's really not until it's connected to the rest of the continuum that we are going to be like making a dent in developing better varieties. And I think there, crop simulation modeling is really a sort of interesting tool to contribute to that, and much of the presentation today is going to be echoing the presentation of David Jordan yesterday, where he talks a lot about that integration between disciplines and approaches. All right, okay, so this is the crop genetic template of a crop model called APSIM. I mean, the only reason why I want to show you that is if grain yield is what we want, eventually, we really have to be clear that it's a number of building blocks behind it. So yield is not a trait, and the focus of my presentation is really going to be on those building blocks of drought adaptation. Second consideration, when we talk about drought, we often think only of what happens when there's drought. And here in that slide, it shows the response of transpiration, which we take as a proxy for growth, for photosynthesis, as a function of soil water. And what it tells us is really a droughted plant really doesn't get into stress until it has depleted at least 60% of the soil water. So if you want to understand what happens in the drought, it's at least equally important to understand what the plants are doing when there's no drought, there's no stress. So it's just want to sort of take you here to the concept of cause and consequences. So certain things that may happen here and the stress which are consequences of what are things that have happened here. And in fact, in much of the trade that we are going to be talking about today, we are talking about those things happening here in the absence of stress. When you talk about climate change and drought, well, we talk about temperature, and for example, the effect of temperature on reproductive success. Well, I like to sort of see the effect and the sort of impact of temperature on the evaporative demand. That's what I've plotted here in that y-axis as a function of time of the year in some of our locations. So that's here in Patancheru and one station we have in Niger. And what you see in that slide is the evaporative demand, what I call sometimes the atmospheric drought is very severe, is very harsh. So I've put that hypothetical sort of threshold above which the plant is really put under stress. I think, I mean, much of the work that we are doing is really on that effect of VPD on the response of crop. It's very relevant for today's work on drought. It's equally important for the work that we do on climate change, because it will have major effect on the plant water balance. What is a drought-tolerant plant? I put that in inverted comma because I think drought tolerance doesn't exist. It's really sort of matter of adapting to conditions. So a drought-tolerant plant for us is a plant that has enough water to fill up the grain, to basically make it to the finish line, but it's a plant that has also depleted all the water that was available to it, with the idea that, well, we don't want to breed for bonsai, we want to maximize the water use. But it gives you an idea of basically the ceiling and the floor within which a crop has to adapt. So what do we have in hand to do is, well, either tap more water, that's the idea of sort of increasing the height of the ceiling, or saving, managing water, or working on use efficiency. So that's basically how does a crop to adapt within those those boundaries. Boundaries which, by the way, are very variable across environments. And so today we're going to be talking about that second part, and our focus is really to understand basically what we call the building blocks of the plant water budget, those causal things, which eventually will turn into interesting phenotype and eventually better yield. So what have we learned so far? We've worked for a number of years in a different level of integration, but I mean, we have used quite a bit, a lysometric system, where the idea is to grow plants in conditions which are as close as possible to the natural conditions. We grow the plant in densities which represent the field conditions. But in those plants, we are not only able to measure yield, but also to measure the plant water budget from beginning to the end of the crop cycle. And so that's a typical sort of water use, cumulated water use, as a function of time after sowing. The stress is applied here between lines that are tolerant and sensitive. And you see that in the green lines, the tolerant tend to extract a bit less water at the vegetative stage, and then they catch up during reproduction and the grain filling. Eventually, they don't differ much for, how much water they extract in total from the soil profile. But it's really the way they manage that limited amount. That's basically the way they move within those ceilings that I was talking about before. So why do they use less water? Well, the two things that we have started working on is look at the canopy conductance, the rate at which a canopy loses water, and then the canopy area. And one of the things that I want to introduce you to is, well, there's other things that we could look at, consequential traits which comes later on during the cropping cycle. That could be sort of manifesting by stay green phenotype or a cooler canopy because the crop is still having water available at that stage of the crop cycle. So more of the same sort of data. That's the case here on pearl millets. It's again water use, not cumulated. It's by week as a function of week after booting. Irrigation is stopped here. Well, very typically, once again, sort of tolerant material tend to extract and burn off a lot more water at those stages, but they run sort of short of it later on, whereas sort of tolerant genotypes are more sort of conservative initially, but then they still have a bit more water here. And so if we do a bit of regression analysis between the water extracted here and a stay green score, for example, that's typically a stay green score that you have, well, we find the same thing in the field, of course. So stay green score is straight related to that water extracted. So stay green is really a consequence of having water available during that week 3 after the stress imposition. And then when we plot the same water uptake in week 3 after booting and grain yields, we've got a very significant relationship with. And then if you extrapolate the yield to a planting density, it comes down to a slope of about 40 kilogram per millimeter of water. And if you look more carefully here we talk only about shifting water from vegetative to reproductive stage in the order of 15 to 20 millimeter. It's not much, but it's a water that makes a tremendous impact on the yield and the stress conditions. So why do plants have different patterns of water use even if they are not under stress? So that's what we have addressed. Different ways, different possible traits, component traits, those causal traits. Of course, differences in the canopy size, no surprise about it. Difference in canopy conductance. And today we are going to be talking about a lot about that part. So we took the case of pearl millet and then we measured that canopy conductance. It's here in the y-axis as a function of the vapor pressure deficit in 2 lines, a tolerant line here and a sensitive one. So there's 2 things. The tolerant line has a lower leaf conductance under sort of mild vapor pressure deficit conditions, and it's also able to further restrict the leaf conductance under high VPD. So it has really 2 merits. The first merit is to save water for later, but the second merit is that because of course this line keeps on fixing carbon, transpiring, but fixing carbon under high vapor pressure deficit is extremely water costly. So basically it's a choice that the crop is making, but for certain conditions it's of course not the right choice. So we tested that phenotype in a number of genetic material here. That's sorghum, where we find large variation between lines. Why is it so important to focus on that trait is when we measure the water use efficiency of those contrasting lines, we see so the transpiration efficiency is measured, well, here again in those tubes. Well, first we've got a large variation, and we find that in fact all those that are VPD insensitive have low TE, and those that are VPD sensitive have a high TE. So that's a phenotype on which we focus, which has really sort of consequence on tangible things. I mean, production of biomass, production of more grain yield per unit of water use. So that's why it's so important. And in fact, at the moment, I guess there's some pioneer groups, people in the room who are testing some of the hybrids because they are behind that same trait in their material for South Asia. So we looked at the same trait in, again, sorghum lines introgress with stay-green QTLs into two different backgrounds. So first with R16, the QTL donor is B35, so here, that's again the leaf conductance as a function of time of the day that we take. We do that in natural conditions. Time of the day is a proxy for VPD, if you do it in the right season. So, well, you see that the donor parent, in fact, has that trait of restricting transpiration. The recurrent doesn't have it. It's there. It's really unrestricted. And we've got two sets of lines. In fact, each of these data points is 4 to 5 lines, introgress with either stay-green 3 or stay-green B. It's actually 3A and 3B. Sorry, I've not sort of modified the slide. They do have that restriction. But the sort of interesting thing is that when you look at that in a different background, so S35 background, there's no difference here. So in other words, the QTL doesn't have the same effect in different backgrounds. And so that's something that we need to be very aware of if we want. I mean, the QTL are basically not magic bullets that would work in every background. So what are the possible mechanisms for that? So I go back to that response. So that again, transpiration rate or conductance as a function of VPD. It's a very quick response. So if you've got a partial stomata closure that goes so quick, it can't be mediated by sort of biochemical signal. I mean, the good old sort of culprit like ABA. It has to be mediated by something else. It has to be mediated by hydraulic signals. And so the hypothesis that we are making is, well, if it's a sort of schematic representation of a plant, where you've got two levels of resistance, here at the leaf and at the roots, resistance to water movement. And the hypothesis that we are sort of looking into at the moment is to see whether there's any sort of hydraulic limitation in the roots of those lines that are VPD sensitive, that make that when the demand for water is too much, basically the plant, the roots can't channel that much water corresponding to the water demand. So that's going a bit more upstream. That's a root cylinder cut. There's two ways for water to penetrate in a root cylinder through the apoplast. So that's basically passive movements through driven by gradients of water potential. And there's then another sort of pathways where the water penetrates plant cells through pores, specialized pores that we call acoporines. And we know that there's differences in the hydraulics of those two pathways. And so the hypothesis that we have made is that, well, maybe VPD sensitive and insensitive line may have different sort of amount of water being channeled through those two pathways. So the way to test that is to sort of measure transpiration responses when we either block the apoplast or whether we inhibit the symplastic pathways. We've got sort of balancers, and the plants are sitting on balancers, and then there's an automatic recording of the transpiration. So in short, and there's a long story behind, just like one slide to encapsulate a bit the data that we obtain. So that's again the transpiration as a function of time when treatment is here. In short, VPD insensitive lines tend to not be that much affected when the apoplast is blocked, but they are a lot more affected than the VPD sensitive line when the symplast is blocked. So the running hypothesis that we are sort of on at the moment is, well, we've got genetic differences in the amount of water that goes through both of those pathways. And of course, the interesting part of the symplastic pathway, which is like sort of held by the aquaporine, is that it gives us sort of a genetic control on an important pathway for water transport. So what we did also was to, well, if aquaporines are involved in those pathways, do we have differences in the gene expression of some of those aquaporine genes? So we picked, I mean, the most contrasting genotypes from sorghum that had either that VPD sensitivity or VPD insensitivity. And we run again an experiment where we measure the response of conductance to VPD. And by collecting tissues, leaf and root tissue here in the morning when everybody is under low VPD. And then we took again a sample in the afternoon. We had two sort of chambers, one where the plant would be exposed to an increasing ladder of VPD, and another chamber where the VPD would be kept low. And so we took again sample here in the afternoon and normalized them against the gene expression of the low VPD chamber to make sure that we don't bang into, I mean, a diurnal effects. And again, to sort of cut the story short, it's quite exciting to see that we've got an over-expression of aquaporine genes, especially in the PIP1 and the PIP2 family, in the VPD insensitive line, the one that sort of, you know, need to channel a bit more water when it is exposed to high VPD. And for me, it's, the exciting part is this one, PIP2-6, because that's also one that's also very exciting in maize. And so I think it opens up a very nice sort of field of work of comparative genomics and, that we have started working and talking about with the pioneer people. But essentially, that's where it's important here to sort of go to the basis, the biological basis of important phenotypes. And I guess we are as close as ever possibly of having genes. I mean, we are still far from knowing exactly what it does functionally, but we are probably as close as we can be to having a gene having a responsibility in a very important phenotype for drought and adaptation. So that was the upstream part. How, you know, with those different traits, I mean, how do we use that into breeding? So we needed a fast way to measure leaf area. We needed a fast way to measure plant transpiration. And so, well, for some of you that are interested, we will have a visit at the Teabreak. Well, the platforms that we have developed is really question-based. We wanted to have a quick assessment of leaf areas. That was the principle. And so we are using a 3D scanning imaging facility where it generates 3D images. And then from those 3D data clouds, I mean, the number of plant parameters are extracted. For example, the leaf area. And so it has a huge throughput and a huge capacity. So that part is fully functional, and we are really now in the measurement sort of mode. The second part, which is up to now in the form of a prototype, is a set of load cells where the plants are sitting on it, and which measure, I mean, constantly the weight of those spots and therefore the transpiration. And so it works really well. We are going to be upgrading the platform in 2015 with more load cells. So with that, we've got really everything in hand of the water budget. The canopy development, something that I call the volumetric growth, and then the transpiration, which is really the transpiration equal photosynthesis, so that's really the massive growth, and we've got that in conjunction. The other thing that we can do with that platform is to measure, I mean, how quick, how large canopy develop. That's basically this idea here, because those differences between sort of different response means the water use for us. The other part that we are going to be able to do over the time is to look at the leaf response to different type of droughts. The atmospheric drought that we've got here, or the soil drought. And so the platform is equipped with, I mean, sensors and all the sets of sensors to be able to capture all the environmental conditions in which the plant are grown and to which they respond. It's basically, it's critical. It's of course critical. And the field application for that would be LiDAR scanning. If I go back to that slide, and I've added a few more sort of possible causal traits for that different pattern of plant water use. And here the possible field application would be to measure stay green and canopy. But again, this idea of cause and consequences is very important, I think, for people that do phenotyping to know where their phenotypes sort of stand, whether they are causal or whether they are consequential. And so it's really important at the time when we talk about field phenotyping. Of course, we need to do field phenotyping for breeding application, but whenever we want to do field phenotyping, it's critical that you know what we measure, because any of the traits that we would be measuring in the field could have many causal relationships. And so if we want to go to the genetic of those causal traits, it's important to know what they are and really sort of be clear with what they mean. All right, okay, so how do we link all those pieces together now? And I think that's where crop simulation modeling sort of is a very powerful tool to sort of put everything back in place and allow us to do prediction on because it's, in the end, it's a trait that we have measured under certain conditions. Does it work? And so it's important to do that. So, while David Jordan yesterday sort of talked about the Australian example, and you're going to sort of see a bit of the same, and in fact, they train us to do that, and thanks. So that's an area of where sorghum is cultivated in South India. That's about 5 million hectares. And so what we did is to take weather data from 14 stations, sort of spread out, and then we run APSIM with that. We calculate the supply-demand ratio. So the supply-demand ratio is the supply is how much every conditions provide in terms of water availability, and the demand is basically what the crop needs on a daily basis. Again, as David said yesterday, I mean, it's extremely diverse. I mean, there's no year similar to the other. There's no location similar to the other. And so that's basically that variability within which we are working. When we cluster those type of response, then we find sort of 5 types: non-stress conditions, very severe early stress conditions here, and then everything in between. And the interesting thing is, if we thought initially that it's a block of areas where sorghum is grown, actually we realize that the predominance of those different type of stress varies in different subregions. And so we've got really 3 subregions coming up, with 2 with, let's say, a fairly mild stress and 1 where the stress is a lot more severe. So it means to say that, well, different type of stress, different type of breeding response, or different type of agronomic management response. So the whole idea of using that stress characterization is really to sort of guide on the decisions that need to be made on each of the, yeah, sure. So once we've got that, we can measure the yield that are in those different regions. So you see that severe stress, I mean poor yield. Here, it tells us that we may have an opportunity to sort of boost up a little bit more the yield by changing maybe the sowing density or certain genetic traits. And once we've got that, then we can simulate the value of some of the traits. So on the yellow one, it's a standard genotype that doesn't have the VPD response, for example. And in the green one, it's a genotype in which we have put in the model that capacity to restrict transpiration and a high VPD, the one that we were talking about before. And so you see that we've got a major yield increase, especially in those environments where the stress is severe. So in other words, if you want to deploy that trait, it's going to be like working really well there. We can also deploy it there, because it will have also an effect, but not that much. So what I mean to say here is, I mean, the prediction that we are making or giving a choice, probabilistic choice for breeders to go for a trait or not to go for a trait. So with that in hand, then we can go back to these integration lines, to those ideotypes, and look at what they've got under the hood. And so we looked at different lines introgressed with stay-green QTL into recurrent background in this one. This line that basically has TE differences, different growth dynamic, and a set of lines in S35 background that have also different sort of ideotype. And the interesting thing is, we see that sometimes the same QTL introgress in different background doesn't touch on the same sort of building blocks of the APSIM template. So that's, again, telling you the complexity. But as long as we can measure those traits, then we know what ideotype is going to be doing. So the next step then is to simulate with those different lines, now that we know what they touch here, what would be their predicted value in terms of yield and stover yield in the different environment. So taking only one example today, it's well, the variability in leaf area. We've seen that some QTL, for example, affect the leaf area in some line in the S35 background. So that's what we measured in the field. So that's leaf area as a function of thermal time. The recurrent is here, and a number of those Teabreak QTL limit and restrict the leaf area and then alter the plant water budget. That's what we simulate with AppSim. That's how it affects the stress response patterns, that SD ratio we talked about before. It basically delays the stress sort of appearance. And here, that's how we represent well, the growth of the biomass, the leaf area, that SD ratio, which falls earlier in the larger canopy line and later in the other one, and eventually the grain yield, which crashes in the large canopy line and the low canopy line. And we can, so in other way, we are able to predict, I mean, what we're going to get in terms of output, not only grains, but also stover. And so that's when the yield gains in different locations. It doesn't work in all the situations, so that gives a sort of probabilistic estimate on where it would work, how many % of the time it's going to work. And I've chosen that example because that's a stover yield gain, and you see that it doesn't work in all the cases. Well, it does not work, basically. It's a trade-off. And so it's important to know those trade-offs because, I mean, there are farmers that may want more stover to feed cattle. Some others may want to sell grains. So what we say is we are not providing solution, we are providing options to the farmers to be chosen. And we can go up to the level of even calculating economic return. And so that's what we calculate in rupees. We've got sort of major return from a larger canopy. So which means that from the larger canopy we lose on the grain side, but we gain on the stover side. And overall, it's positive. So that's, again, to give you the idea of the trade-offs, this time the economic trade-offs. So we do quite a bit of simulation now. That's also the way we can represent it. That's the sort of simulation we do in legumes. And here it's actually a simulation that look at not even a genetic trait, but an agronomic trait. And so the importance of this is, well, you can see where you're going to have the big sort of yield benefit by changing that management way, where it's not going to have much benefit. So it's important also for knowing where to test things on farmers' field, but also to choose testing locations. To summarize, that's how I sort of see the connection. We talked about trait variability and trait dissections. It's important to have in between the crop simulation sort of layer to validate a trait, the value of the trait, and knowing where it's going to be working. That's also where there's sort of an interface with genomics. I mean, once we know it works, I mean, it's the moment to sort of harness the genetic of those critical traits. And of course, linking again all that with field evaluation. Again, the important here, field evaluation, but really connected to what we do on the trait dissection side. So last slide on the key messages. Well, we understand that, well, for drought adapting to water limitation, it's important to manage, water in those in the seeding on the floor. That's where we came to realize the importance of distinguishing between cause and consequence of traits. And that's where it's essential when we talk about a push for field phenotyping. It's very important that we keep a component of trait phenotyping for those critical traits. And then the simulation being there as a tool to sort of stitch the pieces together and to sort of reconstitute a very complex puzzle. Modeling being there to really sort of provide options with the stochastic values on those options that are proposed. So thank you very much.
Thank you, Vincent, for dissecting such a complex trait on water use and transpiration efficiency. I'll take some questions. There's one here.
Hello. Hello, sir. You have showed the estimate for phenotyping. The cost of this estimate is very high because due to that high cost, some of the institutes, they are interested to do the to screen the genotypes such as chickpea as well as other sorghum. Is there any estimate which is having the lower cost, minimum cost? Because we have seen that estimate, phenotyping estimate, on 6th January.
So what you mean, is there anything that can be...
The cost of phenotyping instrument, whatever you have shown.
Oh, you want to know how much it costs?
It is very higher.
Yeah, the question is the cost of phenotyping, so just briefly.
Well, I don't know, in French we say that you don't do omelettes without breaking eggs, and at some point we need to invest in those things. And in the end, if I compare to the cost of some phenotyping platform that exists around the world, it's not that costly. What we have here costs about $750,000. So it's not that costly compared to the millions that have been invested in some platform.
Yeah, there are 3 questions. Please make the questions brief and the answer also brief. You first there.
Hi, this is Debashish from DuPont Pioneer. It was a nice presentation. My question is over here.
Okay.
So you have emphasized on the causal and consequence traits and subtraits. So how do you distinguish between causal and consequence subtraits? Is it through your APSI model or do you have a trait ontology? So how do you do that? Because it's very much context dependent.
Sure. Well, yeah, the model there is a nice tool to see what changes if you change some of those building blocks. There are certain things which are fairly obvious. For example, in that relationship between stay green and the water extraction, well, it's clear that stay green is a consequence of different things. So, the causality, consequentiality of things, it's when we realize that certain sort of integrated traits like stay green, for example, can be explained by different causes. It's one thing I didn't say on those different ideotypes that have different dynamic of leaf development, different TE, different sort of water capture. All those lines, those introgression lines that I've listed there, they all end up with a stay green phenotype in the field. So in other words, here the cause-consequence is very clear. In some cases, it's not going to be that clear. And even all those traits that I've presented on the VPD response, the leaf area, even them, they are consequence of other things. But that's where we are. We need to sort of break them in small pieces that are amenable for genetic analysis in the end. And the work on aquaporine is just one example of that.
Okay, there's one here, and
Hello.
then there, and then last one will be Peter.
Hello, Vadez. I'm Bharadwaj from IRI. See, what as a breeder I'm interested more is yield per se under drought conditions.
Where?
And then all my segregating populations are actually, advanced under normal conditions. As a physiologist, even Neil Turner has suggested transpirational efficiency where later it has become not the cause but the consequence of drought, isn't it? Can you suggest me some traits which under normal selection process or the normal condition process correlate with yield under drought, which will be more beneficial to breeders?
So
the question is basically, what are the traits associated with yield under drought? So just list a few traits.
No. I'm asking what is under normal conditions which I can use as selection indices in the segregating generations.
Well, it's a bit what I was trying to say here. It's this number of traits that we measure in I think what you call normal condition. I get fully irrigated. Maybe that's a chance for me to reiterate that all those traits that we are looking at are measured under fully irrigated conditions. And for example, one of those traits is a capacity, well, that's the one I've talked a lot about today, is the capacity to restrict transpiration under high VPD. That's what's one of them, which you measure under normal conditions, and that basically brings amazing benefits in pretty much all the crops that we have worked on. But I see where you go. You want a magic bullet. There's nothing like this. There's no magic bullet. It's always going to be like context dependent.
We can discuss that at coffee time. Very brief, please, and then
Thanks,
Peter.
thanks, Vincent. That is always nice to be listening to a hardcore plant physiology talk in a genomics conference. Thanks for bringing about
First
this.
time, huh?
It's time. And it's also nice to know that the VPD responses, the transpiration, the stomatal conductances are important. What I wanted to know was, or to comment was, like, have the VPD insensitive individuals, which are actually having some kind of a mechanism to now reduce their temperatures, though not through transpiration, by reflecting radiation. Can that increase the transpiration efficiency and therefore much better adaptability?
Yes, no, very good question, Shesha. It's, yeah, I mean, there's no magic bullet, there's always trade-offs, and indeed, it's, and I'm sure Peter will tackle me on this. I mean, of course, I mean, a plant that restricts transpiration under high evaporative demand, high radiations, warms up, and so there's trade-offs on basically the actual leaf VPD. I mean, the vapor pressure deficit that the surface of the leaf is sort of facing. So indeed, we are going to be losing a bit of the advantage, of the TE advantage from that warming. We know that, but there's no way, but we still believe that the trade-off is less than the benefit. And in fact, when you look at the TE differences that we find, well, it tells us that we kill off the trade-off.
Okay, Peter, your last question.
Yeah, Vincent, good to follow that question, is that absolutely support the approach you're taking, the phenology going into the model, looking at traits. My question and concern is around how you set up the model. I know something about that. So you're saving water before flowering and using after flowering. So you've got to make sure that in the simulation that water is safe.
Yep.
So issues such as weeds, soil evaporation to depth, so those analyses in West Africa, soil evaporation will go down to 1 metre in those soils. If you don't account for that in the model, then you're overestimating. So my question is, what about other approaches? Rather than save the water in the soil, which is at risk from other uses, what about producing carbon, biomass, and looking for genetic variation in translocation of pre-anthesis simulates? So you use the water before anthesis, but you look for variation and how much can be retranslocate.
Sure. No, you're right. I mean, that well, the evaporation sort of part is a key into the question that Shesha was asking. Yes, there's a risk that if you don't use the water now, you run short of it later. About sort of accumulating carbon early. Yes, that's in fact that's the one thing I think we should look at, for example, for legumes. Because in the case of legumes, the case of legumes have a different problem to fix. Well, legume and the drought stress. They need to keep water for the grain filling part. That's typically what you find, for example, in chickpea or cowpeas. But the legume has another problem, is that they need to fix nitrogen. And in legumes, the nitrogen fixation drops usually after flowering. So they need to basically grow and fix carbon so that they can crank in the nitrogen fixation. And so maybe that's one of the strategy to, and in fact, that's the sort of data we find in beans.
I think, Vincent, we
That's
can
exactly
discuss that later, given the time. So please join me in thanking all the speakers this morning.